This video is Unit 2, Learning Target 10, Enzymes. Just as a quick review, when we talk about chemical reactions, when a chemical reaction is creating bonds, energy is being stored up in that bond. When a glucose molecule is created through the process of photosynthesis, energy from the sun is captured and is stored in those bonds. And therefore, when a glucose molecule is consumed, like when you eat a glucose molecule, the energy from those bonds can be released as the molecules are broken down. And this gives energy to body systems. You can store a lot of energy in chemical bonds. Rocket fuel is an example of this. When those bonds are broken through the process of combustion, you can put something into space with that energy. Here's an example of another reaction that releases a lot of energy. When water is split into its individual parts, that releases a lot of energy. The sun is required to actually break those bonds. It requires a lot of energy in order to do that. The energy, in order to get a reaction started, is called the activation energy. So the activation energy is the energy required to begin a chemical reaction. So let's look at this particular graph in terms of activation energy. Activation energy is usually shown with this little E with an A underneath it. Now, what do you see going on in this graph? Whereas, we'll just say that this bottom thing is time, and energy is over here on the right. Well, what's going on the left? Energy is going up, but what happens? So you have the reactants here, the products here. Which one has more energy? The products have more energy, right? And so this is a reaction that actually stores energy. And so look, what do you expect to happen? The activation energy for this reaction is really high. But once you reach that energy of activation, the reaction happens and you get some products. Now, what would you expect the opposite reaction to look like? A reaction that has a reactant, and when that reactant is broken, it releases energy. It's just basically the same graph backwards. Notice the activation energy is actually lower for that. So this would be a reaction like this. Very smaller, or a lot smaller activation energy. And then what happens is the reactants have more energy than the products, and so energy is released as opposed to stored up. Now, these types of reactions are happening all the time in your body. You are storing energy in bonds like ATP, and you're releasing that energy from those bonds over and over again. Now, in order for your body to function well, it needs something called a catalyst. What is a catalyst? A catalyst is something that speeds up a chemical reaction. If I was to leave a piece of paper on my desk for the next couple thousand years and came back and looked at it, I'd be the oldest person alive, but I would also see a piece of paper that was very yellow and brown, maybe even hard to read. What is happening to that piece of paper over the course of years? It is actually burning very slowly over the course of the years. I can use a match to speed up that process. That match would be a catalyst in this case. Well, you don't have matches inside your body, but you do have catalysts in your body. And this is where enzymes come in. An enzyme is a biological catalyst. How do catalysts work in the technical sense? Yes, they speed up reactions, but a catalyst in the technical sense lowers the activation energy of a reaction, therefore increasing the speed and efficiency that reaction can happen. So in your body, an enzyme will take some reactants and make them into the products a lot faster then that would happen, and it does that by lowering the amount of energy that's needed in order to start that reaction. Now, enzymes, let me go back to this. If you'll notice here, we have some helixes and some sheets because enzymes are typically proteins. Enzymes are typically proteins. Remember how we talked about proteins have a particular shape? Well, an enzyme is shaped exactly how it needs to be in order to make that reaction happen. We'll talk about that in a second. Here's a graph, the same kind of graph we looked at earlier, whereas the red line is the reaction without the enzyme, and the black line is the reaction with the enzyme. Notice what happens to the activation energy. It goes down. Therefore, that reaction is going to occur more efficiently. It's going to use less energy, and it's going to happen quicker. So, how does this work? Well, here's the enzyme. We're going to make the enzyme look like some sort of blue shape here. And the enzyme, notice, has some shapes that are specific. It's not a random thing. All right. Now, the substrate in this case are just the reactants in an enzyme reaction. It can be one thing that the enzyme is breaking into, like you see in this reaction, or it can be two things that the enzyme is actually joining together. And when the enzyme and the substrate join together, this is called the enzyme substrate complex. Here's another picture of this. This is where you have two pieces that are joining together to make one piece. It's a chemical reaction. Two things come together to make one new thing. And notice what's happening. Here's the enzyme, and the substrates join together at this place called the active site. So back here on this one, this is the active site. It's the same idea. The active site's there, and those substrates, notice, are particularly shaped for the active site. Now, a good way of explaining an active site is think about a rubber glove. And a rubber glove is shaped like a hand. So it is perfectly, or it's able to you're able to put a hand in it. However, my hand does not perfectly fit inside that glove. The glove actually has to stretch out to fit my hand. And we would say that that is an induced fit. It's definitely designed to put a hand in there, not a foot or a head or something else. But it does kind of stretch out. And the same thing happens with the active site on enzymes. The enzyme kind of forms around the substrate in order to cause the reaction to occur. Now, these two beans, or whatever they are, could float around in your body and eventually react. But this enzyme does a lot better job of actually going out, finding the pieces, and connecting them. And notice what happens. After the reaction occurs, you have a product that's going to go off and do some sort of job or something. And notice the enzyme is unchanged. And so in an enzyme reaction, the substrate will eventually be used up, and the enzyme will be left over. And so if you add more substrate, then that enzyme will go back to work. And we're going to see this in the lab. Now, how can enzymes be regulated? Enzymes can be regulated in a couple ways. First of all, just the amount of substrate. When the substrate is gone, enzymes will no longer react, and they'll just kind of wait around. Enzymes can also be denatured because enzymes are indeed proteins. And so you can do that by changing the temperature or by changing the pH. Enzymes work best at a particular pH. In your stomach, or particular temperature, in your stomach you have a pH of 1. The enzymes that are in your stomach, there's one enzyme called pepsin. It works best at pH 1. If you take and put it in water, it will not work. It will actually denature because it is designed to work at a very low pH. And the enzymes in your body are designed to operate at 98.6 degrees. When you have a fever, your fever is actually designed to destroy bacteria and whatever viruses or things that are going on in your body to denature their proteins, while at the same time keeping yours safe. And so if you have a really high fever for too long, you can actually do brain damage because it will actually begin denaturing the proteins than in your brain. And so enzymes have a very delicate balance in your body and can be regulated by your body in order to regulate its reactions and therefore your body processes. Enzymes are very important for how your body functions.